I will discuss to you about the microbial structure. Bacteria, or in singular bacterium, are classified as prokaryotes, which are single-celled organisms with a simple internal structure that lacks a nucleus and contains DNA that either floats freely in a twisted, thread-like mass called the nucleoid, or in separate circular pieces called plasmids. Ribosomes are the spherical units in the bacterial cell, where proteins are assembled from individual amino acids using the information encoded in ribosomal RNA. Bacterial cells are generally surrounded by two protective coverings, an outer cell wall and an inner cell membrane. Certain bacteria, like the mycoplasmas, so from the word mycoplasma, it is a genus of bacteria that lack a cell wall around their cell membranes. But some bacteria may even have a third outermost protective layer called the capsule. Whip-like extensions often cover the surfaces of bacteria. Long ones called flagella, or short ones called pili or pili, that help bacteria to move around and attach to a host. Um, their classification, um, uh, a few different criteria are used to classify bacteria. The organisms can be distinguished by the nature of their cell walls, by their shape, or by differences in their genetic makeup. The Gram stain is a test used to identify bacteria by the composition of their cell walls. It is named for Hans Christian Gram, who developed the technique in 1884. The test stains positive bacteria, or bacteria that do not have an outer membrane. Gram-negative bacteria don't pick up the stain. For example, Streptococcus pneumonia, which causes pneumonia, is a gram-positive bacteria. But Escherichia coli and Vibrio cholerae, which causes cholera, are gram-negative bacterial. There are three bacterial shapes: round bacteria called pucae, or in singular coccus, and cylindrical capsule-shaped ones known as bacilli, or in singular bacillus, and spiral bacteria, aptly called spirilla, or in singular spirillum. The shapes and configuration of bacteria are often reflected in their names. For example, the milk-curding Lactobacillus acidophilus are bacilli, and pneumonia causing pneumonia are chain of pucae. Some bacteria take other shapes, such as stalk, square, or star. The reproduction, most bacteria multiply by a process called binary fission, according to the life sciences. In this process, a single bacterial cell called the parent makes a copy of its DNA and grows larger by doubling its cellular content. The cell then splits apart, pushing the duplicated material out and creating two identical daughter cells. Some bacterial species, such as cyanobacteria and firmicutes, reproduce via budding. In this case, the daughter cell grows as an offshoot of the parent. It starts off as a small knob, grows same size as its parent, and splits off. The DNA found in parents and offspring after binary fission or budding is exactly the same. Therefore, bacterial cells introduce variation into their genetic material by integrating additional DNA, often from their surroundings, into their genome. This is known as horizontal gene transfer. The resulting genetic variation ensures that bacteria can adapt and survive as their environment changes. There are three ways horizontal gene transfer occurs. First, transformation. Second, transduction. And third, conjugation. First, transformation. It is the most common process of horizontal gene transfer and occurs when short DNA fragments are exchanged between donors and recipients. Then the second is transduction. It involves transfer of DNA from one bacterium into another through bacteriophages. Then the third one is conjugation. Involves transfer of DNA via second phyllos and requires cell-to-cell contact. DNA fragments that contain resistant donors can then make previously susceptible bacteria express resistance as coded by these newly acquired resistance genes. Then, in bacteria in human health and diseases, so the bacteria can be beneficial as well as detrimental to human health. Commensals or friendly bacteria share space and resources within our bodies and tend to be helpful. There are about 10 times more microbial cells than human cells in our bodies. The highest numbers of microbial species are found in the gut, according to microbiologist David A. Rilman's 2012 article in Nature.